If you're not astonished by quantum mechanics, then you haven't understood it. You're meant to be astonished by it. And even quantum physicists who spent their lives thinking about quantum phenomena should be astonished. In fact, if they get very blasé about it, they should take a step back and think about exactly what it means. Now, quantum biology is, well, this image is a picture that appeared in Nature, an article written by the science writer Philip Ball. It introduces some of the speculative ideas where quantum mechanics seems to be playing a role in biology. And to introduce the subject, I could have started from various different points, but I think I will start by saying something, explaining why there's an image of a bird in this picture. This is the European robin. It looks, for those twitchers in the audience, it looks slightly different, and I'm not, so I had to look carefully to see the difference. It looks rather different to our own British robin. The British robin has a deeper red breast than the rather fainter European cousin. The other difference is that the British robin remains here on these shores all year round because we have a reasonably temperate climate. The European robin that lives up in Scandinavia and Russia and Northern Europe obviously would like to find a warmer climate during the winter and migrates down to the Mediterranean, very often down to Spain and even down to the north coast of Africa. And so it follows this path directly from central Sweden, let's say as an example, down to Spain, and it knows how to navigate. Now, this is not new. We've known for a long time that many animals, birds and marine creatures can find their way across very often thousands of miles, even if they've never done that journey before. They use all sorts of tricks. They use the position of the sun. Sometimes they have a celestial map. They know the positions of the stars if they migrate at night. They will recognize local landmarks. Very often they sense the Earth's magnetic field. They have an inbuilt compass. Now, the first person to realize this, so let me say something about the Earth's magnetic field. What was actually quite surprising is that even to this day, scientists don't really understand very well the origins of the Earth's magnetic field. But roughly, it's to do with circular currents in the molten core of our planet that induce magnetic fields due to the rotation of the Earth. So you can think of this giant bar magnet sitting at the very core of our planet and field lines circulating out of the Earth and around. Our magnetic field is very important. We think it formed between 3 and 4 billion years ago when the Earth was very young. It's certainly, without the magnetic field, we wouldn't be alive today, because not only does it shield us from cosmic rays from outer space, it stops those cosmic rays from knocking and blowing away our atmosphere, and therefore water evaporating, as we believe happened on Mars. The first scientist to investigate the influence of the Earth's magnetic field on migrating birds was a Russian zoologist and explorer, Alexander von Middendorf. And he carried out experiments to find out how these birds migrated and what directions they took. They were rather crude experiments, but he postulated that they could sense somehow the presence of the Earth's weak magnetic field with some internal magnetic material that would act as a compass. We know today that many creatures can sense the Earth's magnetic field, from sea turtles to whales and dolphins to obviously many migrating birds to even to the trout. And even research suggests that ants have an ability to sense tiny changes in the magnetic field due to nanocrystals of iron oxide in their antennae. And that's basically the general idea, that you have these crystals of usually magnetite, which are sensitive to the direction of the field, and they can affect electric pulses, signals, currents sent by the neurons to the brain. The homing pigeon very often uses a whole range of different methods to find its way, from remembering landmarks to following the direction of the sun. But also many birds are found to have these tiny magnetite crystals at the ends of their beaks. And what happens is, when the bird is aligned in a particular direction relative to the Earth's magnetic field, then that will cause these magnetic crystals to adjust and move, and as they do, they will create tiny currents and pulses that can be magnified and send signals via the neurons to the brain, signaling to the bird to move in a particular direction. This was discovered, well, the notion that animals, birds, could actually detect the Earth's magnetic field goes back to the mid-19th century. More recently, work carried out in the late 60s and early 70s suggested that there was another way that certain animals, certain birds, could detect the presence of the field. So this is a husband and wife team, the Germans, the Wilczkos, who published controversial work in Science in the early 70s. They carried out an experiment on European robins and realized that the robin certainly could sense the direction of the Earth's magnetic field, but it wasn't doing it in the traditional way that we would use a compass. So these birds weren't sensing which direction was north and which direction was south. Whatever compass they were using didn't depend on the polarity of the field. And they carried out increasingly sophisticated experiments where they would capture these birds and then put sort of artificial magnets outside these chambers where the birds were caught, and reverse north and south of the magnets and see what happens. And the birds didn't seem to be affected by which direction was north, which direction was south. They deduced that the birds weren't following a north-south direction within the field, but rather pole to equator, equator to pole. And they realized that it wasn't the direction of the Earth's magnetic field, but the angle of inclination of the field. Let me explain what I mean. If you think about, so this is the field lines. These aren't physical lines. You remember the experiments at school with iron filings. The iron filings will create the landscape of the magnetic field, showing the direction of the field lines. For the Earth, of course, the field lines come out of the South Pole, or the equivalent to our geographic South Pole, out and around and go in to the North Pole. And so close to the poles, the magnetic field lines are vertical, or almost vertical. But past the equator, they are parallel to the ground. And so depending on how far north or south you are, the angle of inclination of the field changes. And research suggested that the European robin was somehow able to figure out a direction to travel based on the angle of inclination. But it wasn't clear what that mechanism was. So here's a contour map of lines of equal inclination angle. And you can see that, for instance, if I put in the path taken by the European robins, they're moving roughly from central Scandinavia with an inclination angle of about 76 degrees, and there's a change, a reduction in angle of about 20 degrees down to the Mediterranean. The strength of the magnetic field also changes, the intensity. It'll drop from something like just over half a Gauss to just under half a Gauss. But there's no evidence yet that the birds are able to somehow calculate, this is where I am because the intensity of the field is this and the angle of the inclination is this. But somehow they utilize this information to tell them which direction to go. So far, so good. This is no longer really controversial. Many more sophisticated experiments have been carried out since the early 70s to convince scientists that birds are able to do this. What was puzzling, of course, was the mechanism. It was still thought that this maybe had something to do with these metal magnetic crystals, for instance, in the bird's beaks. Because the birds, even the European robin, would utilize various different techniques, even if it followed this rather strange, mysterious method of knowing the angle of inclination of the magnetic field to find its way. It certainly used other more traditional ways to find its way back. For instance, possibly using these magnetic crystals. But how does it find its direction in the first place? The mechanism that was proposed was made by a group at University of California, Irvine, about a decade ago. And it's based on a process that takes place down at the molecular level, something called the radical pair mechanism. So what I'm going to dive in here into some quantum mechanics that is rather complicated. I'll zoom back out again and tell you a bit more about the weirdness of quantum mechanics in a few minutes. So here's the title of their paper, a model for photoreceptor-based magnetoreception in birds. The idea was that whatever the mechanism was going on, it seems that the birds were able to somehow sense through their eyes the direction they should travel through the Earth's magnetic field. And the clue was a particular protein in the photoreceptor cells at the back, on the back wall of the bird's retina, a protein called cryptochrome. Cryptochrome is known to be stimulated by light, particular wavelength of light, typically blue light, that can excite atoms within the protein. And so, which is why cryptochrome had to be in the bird's eye for it to work, because it had to actually have light coming into the eye to get this mechanism working. It's still controversial whether the process takes place in just the bird's right eye or both eyes. It would seem strange that it's only... You'd think evolution would be a bit more, sort of free and easy about which particular eye would be used. Surely birds that could utilize the mechanism in both eyes would have some evolutionary advantage. Nevertheless, here's the quantum mechanics that takes place. Two electrons in an atom can exist in what's called a quantum state. So they have the same energy. But they have to spin in opposite directions. So in a very broad, rather non-accurate quantum mechanical way, we can say one spins clockwise and one spins anti-clockwise. And they form a pair. And if one is spinning clockwise, the other one has to spin anti-clockwise, otherwise it has to go and live somewhere else. This is something called the Pauli exclusion principle, which says that there are rules about how many electrons can be close together in the same state and what direction they spin in. In cryptochrome, it's thought that light, photons, particles of light, will come in and hit and knock one of those electrons in the pair. And that electron will be knocked off into an adjacent molecule, forming what's called a radical pair, molecules that are both electrically charged. One is now positively charged because it's lost an electron, and one is negatively charged because it's gained an electron. That's fine. What's magical quantum mechanically is that although these electrons are far apart, living on different molecules, they still know about each other's existence. If one is spinning one way, the other one doesn't have a choice. They can exist in particular quantum states. The technical jargon is they can exist in what's called a singlet state or a triplet state, depending on whether their spins are aligned, both clockwise or both anti-clockwise, or opposite. Now, quantum entanglement is the idea that particles, however far apart they are, still somehow, their fates remain intertwined. They are still aware of each other's existence. Last year's Nobel Prize for Physics was awarded to the two leaders of two research labs, one in France, one in America, where they've pioneered research within physics, nothing to do with biology, but within the very sort of clean, simple environment of a physics lab where you think you can get hold of and understand these quantum mechanisms. And so quantum entanglement, even for physicists, is a rather novel thing. For it to take place inside these molecules of the cryptochrome protein, inside a cell, at the back of a retina of a bird, and somehow play a role in helping the bird navigate, would seem very, very strange indeed. But these two electrons that are far apart, that are entangled, how they behave and the way they spin affects what chemical reactions those molecules can induce within the cell. Different chemical reactions would send different signals to the bird's brain. The magnetic field outside that the bird is flying through affects these two electrons, these entangled electrons. And so the direction the bird is moving affects the chemical reactions that this radical pair of molecules can cause to take place within the retina. Two things to say about this. First of all, that it's not confirmed that this is the mechanism that takes place. It's the leading candidate to explain how the bird does what it does. It's known that it sees the magnetic field somehow. It's known that it does it somehow through its eyes. It's known that within the eyes there's this protein called cryptochrome that is photosensitive. But we're not sure that this is what takes place. The skepticism arises because quantum entanglement is a very weird and delicate quantum process. The mechanisms that take place in the quantum world disappear very quickly once you make your system complicated. Once you, rather than have just a single atom, you can imagine a single atom. We do experiments like this where you send an atom and it can go in two directions at once. And I'll say something about that in a moment. But the notion that that atom can do this strange quantum behavior of doing two things at once, or electrons doing two things at once, or electrons being somehow connected at a distance, what Albert Einstein said, spooky action at a distance, which is why Einstein didn't like some of the features of quantum mechanics. For that to take place within the warm, complex, messy environment of a living cell was something that most scientists had discounted until recently, because it was deemed to be a process that would just disappear, evaporate, what we call decohere, very, very quickly, far quicker than the biological timescale needed for, there's no point in these electrons, they're entangled for tiny, billions, trillions of a second, and then somehow that's it, the quantum effect has gone. That's no use to the bird. Somehow it's maintaining this quantum behavior for longer. If this is true, if quantum entanglement really does play a role, well, there are other quantum phenomena that go on in the world, in the subatomic world. How many of them play a role in living cells? We know that essentially, let's imagine, the living cell, the human brain, or any organism is made up of cells, and those cells are very complicated, complex cities involving processes involving proteins and enzymes, and there's so much going on. Zoom in, and you can imagine it's sort of a mechanistical world, which is why biologists to this day still feel more comfortable with their balls and sticks models of molecular structure. And it's acknowledged that if you look down deep enough into molecular biology, at its heart, it's essentially organic chemistry. And organic chemistry is, of course, subject to the rules of quantum mechanics, because quantum mechanics tells the atoms, the electrons, how to arrange themselves within atoms, how the atoms fit together to the molecules, and so on as you zoom out. But can those quantum effects somehow leave an imprint on the macroscopic world? Well, it's useful to remind you that living organisms really are the only macroscopic objects whose dynamics are controlled by a single molecule. Very often, a single molecule can affect the whole organism. You can think of this in terms of a mutation in DNA, for instance. A single mutation, a change down at the molecular level, can propagate and have a magnified effect on the whole organism. Until recently, this was deemed to be rather cranky, to be honest. 10 years ago, I don't think I'd even be brave enough to give this talk, because I'd be seen as one of the crackpots and aligned with the many sweet souls who write to me and email me on a regular basis with their new theories of the universe, which I collect, by the way, in a box folder because they're very sweet. But one such cranky idea was around, I guess, 20, 25 years ago, and it was the idea of human consciousness having a quantum origin. The reason it gained any traction at all was because one of the two people involved in proposing this idea was Roger Penrose, one of the greatest mathematical physicists alive in the world today. And Roger Penrose, if you don't know him, he's a cosmologist, and he did a lot of early work in the 70s with Stephen Hawking in developing the mathematics of the Big Bang and the properties of black holes. He teamed up with this other guy, Hameroff, published their ideas about whether we could understand the nature of consciousness through quantum mechanics. The notion that within the neurons in the brain are proteins, microtubules, which can have sort of two different shapes at the same time. You know, in the quantum world, things can do two things at once. So these molecules could be in two different shapes at the same time, and that was fine. That quantum behavior could sort of propagate. If enough of these molecules were playing the quantum game, then it would reach some sort of threshold, and that threshold would collapse the quantum state, and that was deemed to be the switching on of consciousness. And it was very, very fluffy and very hand-wavy and very dodgy. The idea was that quantum mechanics is very, very weird and we don't really understand it. Consciousness is really weird and we don't understand it. The two must be connected. And for a while, there were, in the late 80s and early 90s, there were lots of interdisciplinary workshops and conferences that brought together philosophers and biologists and quantum physicists and computer scientists and so on, because this was seen to be the new big thing, and it sort of died down. So if we are going to see quantum effects in biology, I think we have to step back and try not to be so ambitious and look a bit more carefully. But I want to remind you of just how weird the quantum world is. And I'm going to explain to you what's known as the central mystery of quantum mechanics. It was Richard Feynman, the American physicist, said, this is the central mystery of quantum mechanics. There's lots of weird stuff that goes on in the quantum world. Hit you with this, and it basically tells you what it's all about. It's called the Two-Slit Experiment. I'll start with this. Imagine you have a source of light shining against a screen with two slits. Now, for the pedants in the audience, this source of light has to be monochromatic light of a particular wavelength, whereas, of course, a light bulb is white light, and that's made up of all the colors of the spectrum, lots of different wavelengths. But imagine this is just a single wavelength of light, and you can see the light is coming out in waves like ripples in a pond. That's the nature of wave-like behavior. As the light hits the screen, it squeezes through the two slits, and each slit in turn on the other side becomes almost like a new source of light, and the light spreads out, it diffracts. And as the waves of light overlap, they will interfere with each other. So where a crest hits a trough, they will cancel. Where a crest hits a crest, they will amplify, and so on. And so on the back screen, you end up with what's called an interference pattern, a series of light and dark fringes where the waves have either cancelled out or worked together in phase. That's fine. That's not quantum mechanics. That's a property of light that goes back over 200 years that we've known about since the early 19th century. Imagine doing the same experiment again, but doing it not with waves, but with particles. Do it with grains of sand. So this is the same experiment, but I've tipped it 90 degrees. Rather than waves that are spread out that wash up against the two slits and squeeze through, here you've got individual particles of sand, and each particle will either go through one slit or the other. And so you see they will sort of drain through, and you get two bumps underneath each of the slits. So the two peaks is reminiscent of particle-like behavior, whereas the multiple pattern of interference is wave-like behavior. What if we do the same experiment with atoms? Well, so imagine we have an atom gun, something that can fire atoms, a stream of atoms. You can't see them because they're very small. Let's block off one of the two slits. So these two slits are the dimensions and separation of the slits is chosen appropriately to show us how atoms do things. And so far so good. Nothing strange here. You'll see a lot of atoms hitting the back screen. So this will now have to be some sort of photosensitive screen whereby when an atom hits it, it'll give off a little flash of light to say the atom has arrived here. So the atoms are arriving as these little pinpricks of light that we see. Of course, a lot of the atoms will be blocked by the first screen. They won't go through that slit. But those that do get through to the other side, and you can see there's a bit of spreading of the atoms. But if we didn't know anything about atoms, we'd say, well, that's fine, we can understand that. A lot of the atoms are going clean through the slit. Some are sort of maybe bouncing off the edge of the slit, and so they're sort of being deflected a bit, which is why you get a bit of a spread. The first mystery of quantum mechanics comes when we open the second slit. Because now we see something that's very much like the interference pattern we got with light. Rather than having two bands of spots where the atoms have gone through the two slits, it's as though the atoms have gone through the slits behaving like waves, and you get interference of the waves and you get these bands. If we know nothing about atoms or quantum mechanics, you could try and rationalize it and say, well, maybe atoms behave in a very strange way, and only a certain number of them are allowed to all sit together. And so, me and my gang, we're all going to go on this slit. No, sorry, no room for you. You go to the next slit above. And by the way, there's this rule that no one can go in between the two bands, but a few naughty atoms do. So there's a bit of a scatter. There could be some forces between atoms that make them coordinate their actions in a way to give this pattern. That's not mysterious. We just don't know how atoms do things. But we can be clever, and we can force the issue. What if we were to not send the atoms all through at once, but send them through one at a time? Leave enough of a gap for the atom to get through to hit the screen. Of course, as I say, some atoms will hit the first screen and not get through, but those that get through will hit the back screen. So let's run the experiment again slowly, and gradually you'll see as the atoms go through, they'll look like they're just randomly arriving on the other side. You keep sending atoms through one at a time, and gradually that same pattern appears. So each atom by itself is somehow contributing its small part to the overall wave-like behavior that we see in the interference pattern. How does it do it? How does... We know the atom's a tiny, localized particle. We can't see it. It's too small to even see under a microscope. We're firing it at the screen with the two slits. Some moment later, you see a flash of light on the back screen. It's arrived in a localized point. It's not spread itself out. You don't get sort of like a wash of a faint light across the whole screen. It's a little point. The atom is localized. It's arrived in a certain location. And yet, it somehow seems to have been aware of there being two slits, not one, because it's given rise to this interference pattern. How does one atom do that? Does it split in half? Does it become like a cloud that goes through both? Well, we can try and be even cleverer. What if we were to spy on the atom and see where it goes? We can gently just observe which slit it goes through. So you put a detector just above the upper slit that will flash or beep whenever it sees an atom go through that top slit. Sure enough, you fire the atoms through, one at a time. 50% of the time, the detector will beep. The other 50% of the time it doesn't, the assumption being that the atom has gone through the lower slit. But of course, I've been cheeky here. I haven't shown you the results of the experiment. That's what you get. 50% of the time, it beeps, and you see a spot arrive adjacent to the upper slit. The other half of the time, it doesn't beep, but you see a spot arrive at the lower slit. So, yeah, it's picked out the atoms that have gone through the upper slit, and not the ones that have gone through. So each atom does go through one slit or the other. But that's a different result to what we had earlier. So here's the last bit of sneakiness that we can play with atoms. Surely now, we're going to get to grips with it. Leave the detector there, but just very quietly go and unplug it. Don't let the atoms know that you're not spying on them.